Today we will learn about major histocompatibility complex, in short MHC. So let's begin. Antigens are presented to T cells by two classes of major histocompatibility complex, in short MHC proteins. T cells recognize antigens that originate within other cells within other cells, such as viral peptides from infected cells. They do not do this by binding specifically to antigenic peptides presented on the surface of the infected cells by molecules encoded by the major histocompatibility complex. The T cells use their specific receptor, in short TCRS, to recognize the unique combination of MHC molecule plus antigenic peptide. Unlike B cells, which recognize just a portion of antigen, a T cell recognizes residues from both the MHC molecule and the antigen peptide. Genetic analysis revealed that the rejection occurs when tissues are transplanted between individuals having different genes in a chromosomal region called the major histocompatibility complex MHC. This large region, that means 3000 kilobase pair, encodes three classes of transmembrane proteins. Class 1 protein present foreign epitopes to cytotoxic T cells. Class 2 proteins present them to help T cells, and class 3 proteins are components of complement cascade. Class 1 proteins are present on nearly all cells, whereas class 2 proteins are confined to cells of the immune system and phagocytes. MSC proteins are highly variable. Now, let's know about the MHC class 1 molecules. Class 1 proteins consist of alpha 44 kilodalton, a chain non-covalently bound to a 12 kD polypeptide called beta 2 microglobulin. The alpha chain has three extracellular domains, that is alpha 1, alpha 2 and alpha 3, a transmembrane segment and a cytosolic tail. Differences between class 1 protein are located mainly in the alpha 1 and alpha 2 domains. The alpha domain, which interacts with a constant beta 2 microglobulin, is largely conserved. Now let's study about the MSC class 2 molecules. Class 2 proteins have a similar overall plan. They consist of 35 kilodalton Kd alpha chain and a non-covalent bound 29 Kd beta chain. Each contains two extracellular domains, a transmembrane segment, and a short cytosolic tail. Class 1 and class 2 proteins bind peptides derived from foreign proteins rather than the intact proteins themselves. The foreign protein is internalized and digested in lysosomes. Some of the resulting peptides are transported to the plasma membrane in association with either a class 1 or 2 protein. Only peptide having an appropriate sequence called a agriotope can bind to a particular MSC protein. For example, peptides capable of binding to ED, a class 2 protein, have the consequent sequence, whereas X denotes a variable residue. An interesting finding is that this consciousness sequence is present in the variable region of MSC molecule capable of binding it. In other words, the MHC protein contains an internal agritope. Thus, peptides capable of being presented by an MHC protein are homologous to a portion of MHC protein itself. The three-dimensional structure of a human MHC class 1 protein, that is in short HLA-A2, has recently been solved by Don Wille, Jack Strominger, and their workers. The antigen binding site is formed by the alpha 1 and alpha 2 domains, which are very similar to the to one another and different from antibody domains. The binding site is a deep groove 25 angstrom long and 10 angstrom wide, which runs between two long helices of alpha 1 and alpha 2. Alpha 3, the alpha 3 and beta 2 microglobulin domains resemble antibody domains but are paired differently. Finally finished major histocompatibility complex. Any questions regarding this you can ask me in the comment box and subscribe my channel to know more updates. Thank you.